Good evening, students. Is the screen visible? Okay. Good evening, dear students. Welcome back. So, in the previous class, we had started with biomolecules chapter, right? So, so this particular chapter, okay, that is biomolecule. This is for 9 marks. Okay. So for 9 marks, this particular chapter is allotted. So for 9 marks, what all are the things you are going to get? So for 1 mark, you are going to get two questions. Okay, MCQs, two questions. Then 2 marks, one question, and 5 marks. That is in long questions. Okay, long question. That in the last part you will be getting 5 marks, one questions. Okay, so that is this is totally for 9 marks. Fine. So in the previous session we have seen what are biomolecules. Right, we have studied in short wise. Okay, all the different this small definitions. Okay, and then we had completed this MCQs. So we have completed the MCQs part also. So when we were solving the MCQs, we have seen what are the different biomolecules which are very essential for our body. Okay, so we have studied the concepts. Okay, so we have seen what are carbohydrates. So in the carbohydrates, we had studied about the glucose structure elucidation part. So this is very important, the structure elucidation of glucose. Okay, so please study the structure elucidation of the glucose. Then okay, so the types of this classification of carbohydrates. Okay, so these things are very important. Okay, so the types of carbohydrates, then we have seen the structural elucidation as I have told you. Okay, structural elucidation of glucose, preparation of glucose. Then cyclic structure of glucose is very important. Then fructose cyclic structure, the Havort structures. Okay, Havort structure. Please study the structures. That is, Havort structures are very important for exam. Okay, so you have to practice this particular structures. That is Havort structure of fructose, sucrose. Okay, glucose. So these particular diagrams you need to the structures you need to study. Okay, so the importance of glycogen. Okay, then cellulose. Okay, so these things. Then we have seen what are proteins. Okay, amino acids, the classification of amino acids. Right, what is peptide bond, polypeptide bond? So this we have seen. Then a small part of vitamins. Okay, different vitamins and their deficiency. Okay, so these were the MCQs, okay, which we have solved in our previous class. So today we are going to see the uh, questions. Okay, questions. So these are some of the important repeatedly asked questions, so which I have taken. Okay, so please answer them. So we'll see one by one. Okay, so the first question. Okay, so the first question is, what are reducing sugar? Okay, what are reducing sugar? uh, what are non-reducing sugar, and you have to give an example of each. Yes, what are reducing sugars? What are reducing sugars? Non-reducing sugars, give an example. What are reducing sugars, students? What do you mean by reducing sugars? Do you know what is reducing sugar? Can you give me an example? Okay. don't explain me. Can you give me example of reducing sugars and non-reducing sugars? Best example of a reducing sugar. Okay. Yes. Somebody has answered. Glucose. What is glucose? Reducing sugar or non-reducing? Reducing. Okay, fine. So what are reducing sugar? Okay. So reducing sugars are those which reduce tolerance reagent and Felling's solution. Right? You know this? Yes. So why it reduces this tolerance reagent and Felling solution, students? Yes. Why it tolerance reagent and Felling solution? So it has a they have a anomeric carbon, okay, because the anomeric carbon has a OH group. So this anomeric carbon, okay, it has an anomeric carbon. Anomeric carbon means what? Where the OH group can be on the right hand side or the left hand side, okay. So the anomeric carbon, because it has an anomeric carbon, so it can reduce the tolerance reagent and Felling solution, okay. Now, what is non-reducing? Okay, before that, what is the examples of reducing sugars? Okay, so the examples of reducing sugar is it is glucose, galactose, maltose, and lactose. Okay, so these are the examples of reducing sugars. Okay, now what is non-reducing sugars? Coming back to non-reducing sugar, non-reducing sugars are those, okay, which do not reduce Tolling's reagent. Okay, do not reduce Tolling reagent and Fellung solution. So the best examples of reducing non-reducing sugar is what? It is what is non-reducing sugar? Sucrose. Okay, remember this, very important. So non-reducing sugar, the best example is sucrose. Okay, it is sucrose. Fine. So write a chemical reaction. Next is write a chemical reaction to elucidate 6 carbon atoms in a glucose which are in straight chain. Then the presence of carbonyl group. Third one, presence of aldehydic functional group. And fourth one, the no. Okay, only 3 points. Okay, so this is the particular question. Okay, so please answer this. So write the chemical reaction to elucidate. So which reaction will you elucidate for this particular, that is 6 carbon atoms in a glucose in a straight chain? Yes. Yes, good. Okay, good. So the reaction with HI, good, good. So the reaction with hydrogen iodide. Okay, so when you react your, that is glucose. Okay, so when you react your glucose with HI, okay, that is when you heat it for a longer time, when you heat it for a longer time, what will happen? It will convert it into N-hexane. Okay, I'll write the reaction here. So I'll elucidate this particular reaction. It's getting just a second. So here I'll write the reaction. So that is CHO. So I'll take it here. So that is CHO, then CHOH 4 times. Okay, I'll write it the shorter way, CH2OH. So when I heat this in the presence of I get N-hexane. Okay, that is N-hexane. N-hexane means what? Carbon atoms are 6. Okay, carbon atoms are 6. Fine. That is CH3, CH2, 4 times, and then CH3. So this is N-hexane. So this is the first reaction where we are showing the presence of 6 carbon atoms. Okay. Next is the presence of carbonyl group. So presence of carbonyl group can be shown by reacting it with ammonium hydroxide. Okay, by hydroxylamine. Okay, by reacting it with hydroxylamine. So the presence of carbonyl group can be elucidated by hydroxyl group. Okay, that is a hydroxylamine. So what it forms? It forms CH double bond N OH, then CHOH 4 times. Okay, and CH2OH. Fine. So this is the reaction. This is the reaction. Okay. So what it is forming? It is forming an oxime. Oxime formation is shown. That is the presence of carbonyl group can be determined like this. Next is presence of aldehydic functional group. Presence of aldehydic functional group, how it is shown? By treating it with bromine water. By treating it with bromine water. Okay. So by treating with bromine water, what you get? You get gluconic acid. You get what? You get gluconic acid. So this confirms the presence of aldehydic group. So one of the, that is your aldehydic group is converted into acid. So it is converted into gluconic acid. Fine. So these are the 3 reactions. Okay. So these are the chemical reactions which elucidate the presence of 6 carbon atoms. Okay. In a straight chain by treating it with hydrogen iodide, you get N-hexane. Then with hydroxylamine, okay, hydroxylamine, you get a oxime, which shows you the presence of carbonyl group. And when you treat it with bromine water, it confirms you the presence of a aldehydic group, giving you a gluconic acid. Okay, so this is the particular reaction. I hope this is clear. Yes, students, is it clear? Is it clear, students? Okay, so moving further, so write the Hayward structure. So as I have told you, so please write the Hayward structure. So please study this. Okay, I won't be writing these structures. So please note this down. So as I have told in my previous classes also, please carry a paper and a pen and make the notes of this. Okay, please note down this important questions because these are very important. Okay, so please write down this particular third question. That is, write the Hayward structure. Okay, very important. Please note this down. I'll give you 2 minutes. Okay, note this down. Write the Hayward structure of alpha D glucose. Okay, then beta D glucose, alpha D fructose, beta D fructose, maltose, sucrose, and lactose. Please note this down. Yes, I hope you have noted it down. Okay. So moving further, next, what are fibrous proteins? Give an example. Yes, students, what are fibrous proteins? And give an example. Yes, what are fibrous proteins, students? So the proteins which have thread-like structure, okay, the proteins which are having thread-like structures, they are called as fibrous proteins. Can you give me example of fibrous proteins? Yes, can you give me example of fibrous protein, students? Yes, okay, somebody has. Yes, keratin. Yes, keratin, myosin. Okay, keratin, myosin. Yes, good. They are the examples. Next, what are globular proteins? What are globular proteins? Give an example. Yes, what are globular proteins? Give an example. Okay, hemoglobin. Fine, good. Yes, hemoglobin is example. So, which are having. Okay, so the proteins which are having spherical structure, the proteins which are having spherical structure, they are called as what? They are called as globular proteins. What they are called as? They are called as globular proteins. Okay, so what is the example? The best example is insulin or hemoglobin. What is the best example? Insulin or hemoglobin. Okay, please remember this. What is the best example? Insulin and hemoglobin. Do not forget the examples. Okay, examples are very important. Okay. Then, so please remember, don't get confused between what is fibrous protein, what is globular protein. Okay, so what are globular proteins? So globular proteins are having spherical structure. Okay, they are having spherical structure, and fibrous protein, they are having thread-like structure. They are having what? They are having thread-like structure. Fine. Okay, next, what is Zwitter ion? Writes its formula. Okay, very important, students, very important. Okay, what is Zwitter ion? What is Zwitter ion? What is Zwitter ion? And write its formula. Yes, students, what is Zwitter ion? Very important. What is Zwitter ion? Okay, so in aqueous solution, okay, when you have, so this is the amide. Okay, so this is the amino acid. How do you represent amino acid? Okay, CH. So this is CH, then NH2 and COOH. Okay, COH. So when in aqueous solution, when amino acid, when they exist as dipolar ion, when they exist as what? When they exist as a dipolar ion. So how they exist as dipolar ion? That is RC. Okay, CH, C double bond O minus and NH3 plus. Okay, so this is the formula. So this is the formula. So general formula, how you represent your alpha amino acid is this. Okay, but the Zwitter ion, that is when you add it in water, okay, when you add it in aqueous solution, it will form a dipolar ion. A dipolar ion is formed. So this is the formula. So this is a formula. So what is Zwitter ion? In aqueous solution, okay, the amino acid exists as a dipolar ion. Then it is called as a Zwitter ion. They are called as what? Zwitter ion. Yes. Is it clear, students? Okay. So please note this down. Very important questions. Okay. Okay. Next is, what are peptide bonds? How they are found? Yes, students. What are peptide bonds and how they are found? What are peptide bonds and how they are found? Yes. What are peptide bonds, students? Peptide bonds means what? How they are found? So the bonds which are found by condensation of amino acid. Please note it down. Bond which is found by condensation of amino acid with the elimination of water molecule. Okay. So whenever the amino acid is getting condensed with another amino acid, suppose NH2, okay, CH2, C double bond OH. So this is one amino acid. when it is getting condensed with another amino acid. So this is another amino acid. Okay. So what is happening here? So there will be this condensation. Okay. There will be water molecule loss. So this will be one hydrogen is lost. Okay. So this will form what? NH2, CH2, CO, NH, CH, then COOH, and CH3. Okay, sorry, I'll write it here, CH3. So this is the amide linkage. Okay, so when two amino acids are getting combined, okay, one peptide bond is formed. Fine. So what are peptide bonds? So bond which is formed by the condensation of amino acid with the elimination of water molecule. So how they are formed? By elimination of water molecule. Okay. So this is the explanation you are supposed to give. Fine. Next is, what are essential amino acids, and give an example. Yes, students, what are essential amino acids? Give an example. What are essential amino acids? Yes, do you know what are essential amino acids? Essential amino acids, they cannot be synthesized in our body. Okay, so the amino acids which cannot be synthesized in our body, and they must be taken through diet. Okay, they should be taken through diet. Okay, that is along with the diet we have to take this. Okay, so the example for essential amino acid is lysine. The example for essential amino acid is what? Lysine. Okay. So what are essential amino acids? So these cannot be synthesized in our body, but they have to be taken through our diet. Okay. So they are called as what? They are called as essential amino acid. Next is non-essential amino acid. Non-essential amino acids are synthesized in our body. Okay. They are synthesized in our body. Okay. So what are the example? The example, okay, glycine. Please remember this, glycine. So non-essential amino acid, they are synthesized in our body. Okay, so they are called as non-essential amino acid. I hope we have solved 10 questions. Is it clear? All the 10 questions are clear, students? Yes. Are the 10 questions clear? Shall we move further? Or if you have any doubts, please ask me here itself. Now itself, you can ask the doubts. Yes. Eh, Any doubts, students? Okay. So we'll move further. The next question is, what is meant by denaturation of proteins? Yes, what is meant by denaturation of proteins? Which level of structure remains intact during denaturation? So this is a question. What is meant? What do you mean by denaturation of proteins? Very important. So the process by which protein lose their biological activity. Okay. The proteins, the process by which the proteins, they lose their biological activity. Okay, when they are subjected to temperature. Okay, when they are subjected to change in temperature or change in pH, then they are called as denaturation of proteins. Okay, so whenever the proteins, they are heated, maybe there is change in temperature, or maybe there is change in pH. So what will happen? These proteins will lose their identity. They will lose their biological activity. Okay. So then it is called as what? Denaturation. Denaturation. Okay. So this is the denaturation. Now, which level of structure remains intact during denaturation of globular protein? So primary structure. Okay. So the primary structure it will remain intact. Okay, it remains intact during the denaturation of globular protein. So what will be the best example? Coagulation of egg albumin on heating. So you have seen egg, right? So the egg will be how it will be if you break the egg. Okay, so the yolk and okay, so the yolk and the white protein part, okay, albumin, it comes out. Okay, but if you boil it for a longer time, what will happen? The egg gets coagulated. It gets coagulated. So this coagulation process, okay, it becomes hard. So from a soft egg, it turns into a hard egg, right? The shell. So so the like that. So this is the denaturation process. So denaturation means what? It changes the biological activity when it is subjected to change in temperature or change in pH. So the primary structure will remain intact, and the best example is coagulation of egg albumin on heating. Okay, so remember this. Next question. Name a vitamin that is stored in liver and adipose tissues or fat soluble vitamin. Yes, students, please name a vitamin that is stored in liver and adipose tissues or fat soluble vitamin. So you have to mention the vitamin that is stored in liver, okay, and adipose tissue, or it is also called as the fat soluble vitamin. Yes, which is the vitamin, student? Yes, which is the vitamin which is fat soluble? It is stored in liver and adipose tissues. Okay. So the fat soluble vitamin. So this can be A, D, E, K. Okay, any one of this. A, D, E, K. A, D, E, K. So fat soluble vitamin. Okay, so any of this, you can write a vitamin which is stored in liver and adipose tissues, or the fat soluble vitamin. It is A, D, E, K. Okay. Now mention one water soluble vitamin. So can you name any water soluble vitamin? Which is a water soluble vitamin? It is vitamin C and B. Okay, vitamin C and B, except your B12, except your B12, vitamin C and vitamin B. Okay, all the vitamin B. So these are the water soluble vitamin. Fine. Next, name the disease caused by the deficiency of vitamin A, B1, B2, B6, B12, C, C, E, D, K. Yes. We'll see one by one. Okay, so these deficiencies are very important. Vitamin A. What does vitamin A causes? Yes, you can give the answer. What does vitamin A causes? Yes, night blindness. Good. Priyanka, Vedvati have answered it right. It causes night blindness. Good. Okay, it causes what? It causes night blindness. Good. Vitamin A causes night blindness. Fine. Then vitamin B1. What does vitamin B1 causes? Yes, it causes beriberi. It causes beriberi. Fine. Vitamin B2, it causes chlorosis. Okay, it causes chlorosis. Yes, CEILOSIS. Fine. Vitamin B6, convergence. Okay, convergence. So convergence are caused by vitamin B6. Okay, B12, pernicious anemia. Okay, pernicious anemia. Amia. Fine. Good. Yes, RBC deficiency. It is also called as RBC deficiency. C, vitamin C, scurvy. Okay, it is scurvy. Fine. Vitamin E, muscular weakness. Okay, vitamin E, causes muscular weakness. Muscular weakness. Okay. Then vitamin D causes rickets. Vitamin D causes rickets. Then vitamin D, K causes delayed blood clotting time. Okay, delayed blood clotting time. So these are the deficiencies. Yes, good. So most of you have answered it right. Good. Okay. Yes, fine. So these are the diseases. So you need to remember the vitamin. Okay, what is the disease which is caused by the each vitamins. Okay, so remember this is very important. Okay, this can be asked in the applicative part also, like in the MCQs also. Okay, so please remember this. Next, which is the nitrogenous base present in only RNA and only in DNA. Okay, so which is the nitrogenous base which is present in DNA and RNA. So which is present in RNA, students? In RNA, what is present? Okay, first we'll see DNA or RNA. Okay, first you tell about DNA. Okay, uracil. Okay, she has told about RNA. Fine. So which are the nitrogenous bases in RNA? It is adenine, then guanine, then cytosine and uracil. Okay, so these are the nitrogenous bases in RNA. Fine. Now DNA, adenine, guanine, cytosine and thymine. Okay, so yes. So these are the bases which are present. Good. Yes. Vedvati, yes. Good. Fine. Next. Which hormone regulates blood sugar level? And second, contains iodine. So which particular hormone regulates the blood sugar level? Which is a particular hormone which regulates the blood sugar level? Okay, insulin. Yes, insulin and glucogen. Okay, glucogen also. Fine. Then which contains iodine? Which contains iodine? Contains iodine. Which is the hormone which contains iodine, students? It is thyroxine. Okay, remember? It is thyroxine. Okay, it is thyroxine. I'll write it down. Thyroxine. Okay. Then steroid hormones. Which are the steroid hormones? Which are the steroid hormones? Steroid hormones. The steroid hormones are estrogens and androgens. Okay, estrogens and androgens. So which are the steroid hormones? They are estrogens and androgens. Okay, fine. Then which are the polypeptide hormones? Which are the polypeptide hormones, students? The polypeptide hormones are, yes, insulin and endorphins. Okay, insulin and endorphins. Okay, so the hormone which regulates blood sugar level, they are insulin and glucagon. Okay, it is glucagon, but the hormone which are polypeptide hormone, they are insulin and endorphins. Okay, they are endorphins. Fine. Then, what is glycosidic linkage? Yes, what is glycosidic linkage, students? What is glycosidic linkage? What is glycosidic linkage, students? Glycosides linkage is what? The linkage, it is between the two monosaccharide units. Okay, so the linkage which is present between two monosaccharide units through the oxygen atom. Okay, it is called as the glycosidic linkage. Okay, it is called as what? Glycosidic linkage. Is it clear? Yes, is it clear, students? So whatever the questions, so these were the questions which are to be dealt in this particular chapter, that is biomolecules. Any doubts? Yes, any doubts? So I'll be asking, okay, some questions. So you can answer them in the chat box. Okay. So we'll take few more questions. So please answer to the in the chat box so that I'll understand that you have how much you have understood this particular chapter. Okay, fine. So we'll see who will answer first. Okay, so you have to give an example for aldohexose and ketohexose. Yes, so give an example for aldohexose and a ketohexose. Yes, let me see who will answer first. Give an example for aldohexose and ketohexose. Nobody? Nobody knows the answer? Yes, students, what is the example? Okay, glucose. What is glucose? Aldohexose or ketohexose? Aldose is example. Aldohexose. Glucose is example of aldohexose. Okay. Then what is the example for ketohexose? Means what? Hexose means a carbon molecule. Aldo means a aldehyde group. Okay. Similarly, ketohexose means a six carbon molecule with a ketone group. Okay. Yes. So the example for aldohexose is glucose, because glucose is having a aldehyde group, and ketohexose, the example is fructose. Okay. Good. Vedvati has answered it right. Good. Okay. So the sugar which is present in milk. Can you tell me which is the sugar present in milk? Yes, students. Yes, Priyanka has answered it right. Good. It is lactose. Good. It is answer. It is lactose. Others also please answer. Okay. The other students also please answer. Fine. Okay. So can you tell me which is the monomer present in starch or glycogen? Which is the monomer present in starch and glycogen or and glycogen? Yes. Okay. Let me see who is answering first. Yes, students. Which is the monomer present in starch and glycogen? No, I want the perfect one. No, not glucose. Okay, what glucose? Monomer. What type of glucose? Yes, Shashank, what type of glucose? Yes, good. Deepak has answered it right. It is alpha D glucose. Okay, yes. Vedvati has given the proper way. It is alpha D glucose. Yes. Good. Okay, good. Fine. Next one, which is the water soluble component of starch? Which is the water soluble component of starch? Which is the water soluble component of starch? Yes, Vedvati has answered it right. It is amylose. Yes, good. It is amylose. Good. Then, which is a water insoluble component? Which is a water insoluble component? Yes, Vedvati, amylopeptine. What? Why only Vedvati is answering? What about others? Yes, she is leading you all. Yes, Priyanka. Yes, good. Okay. So be little fast next time. Fine. Okay. We'll next one. So suppose, okay, I'll give you an example. Now you know what is polypeptide. Okay, what is a peptide bond? What is a polypeptide? Okay. Now I have a pentapeptide. Okay, I have a pentapeptide. Peptide. So how many bonds are there? How many bonds are present in pentapeptide bond? Yes, Vedvati has answered it right. It is four. Yes, Shashank has also answered it right. It is four. Okay. So okay. So when a peptide bond is formed, so pentapeptide means five, these are there. Okay. Five amino acids are there. Okay. But it will form only four peptide bonds between them. Okay. Now, suppose I have a tetrapeptide. Tetra means four. So how many peptide bonds will be formed? Three. Yes, Shashank has answered it right. Yes, Priyanka has also answered it right. It is three. Good. Okay. Now, can you tell me the chemical composition of nucleic acid? Can anyone tell me the chemical composition of nucleic acid? What does a nucleic acid composed of? What does the nucleic acid composed of? No. What is the chemical composition? They are the types of nucleic acid. Okay, you have answered a type of, yes, Priyanka, yes, you are a bit closer. Yes, nitrogenous base, pentose sugar, and nitrogenous base, pentose sugar, and phosphoric acid. Good. Yes, Priyanka has answered it right. Vedvati have also answered it right. Okay, good. So a pentose sugar, a phosphoric acid, and a nitrogenous base. Good. Fine. So which is a nucleic acid which synthesize protein? Which is the nucleic acid which synthesize protein? Yes, Sashank has answered it right. It is RNA. Good. And which is the nucleic acid uh, which which is responsible for genetic information? It is DNA. Yes, good. Sashank has answered it right. Okay. So which is the sugar which is present in RNA? Which sugar is present in RNA? Which sugar is present in RNA? Yes. Beta D ribose. Yes, Shashank has answered it right. It is beta D ribose. And in DNA, which sugar is present? In DNA, which sugar is present? It is beta D. Yes, 2-deoxyribose. Yes, Priyanka has answered it right. Good. Okay, fine. Okay. So this completes the chapter biomolecule. Okay. So did you understand the concepts? Did you understand the concept? So whatever the things are told, did you understand them? Yes. Okay, fine. Any doubts? If you have any doubts, you can ask. Did you enjoy the session? Okay. Are you preparing for the exams? Are you studying? Are you all studying? See, for 5 marks question, okay, 5 mark question in your what is that? Organic chemistry. Okay, so all the long questions are from organic chemistry, and 5 marks question directly they won't be asked only. 5 mark question they won't be asked. Either it will be 2+3 or 2+2+1. Okay, so 5 marks question is never asked directly. So it can be 2+3 or 2+2+1. So we have covered all those, right? We have covered all those questions. So whatever has been told, so these are the important things. Okay, and I have covered, right? So, okay, fine. Yes, this is the blueprint which is set by the Pew board for the academic year 24, okay, 23-24. So according to this particular blueprint only, you will be getting the paper, okay, for your board exams. So this blueprint is already set. So this is according to the Pew board itself. Okay, so this is a confirmed blueprint. Yes. Okay, fine. So no other doubts. Yes, I hope you all are studying. Okay, do not, whatever the things are told, please make the point of them. Okay, and study them. So it will it will you will get some uh, extra marks. Okay, you will get some extra marks. So okay, so these are some of the important questions. So most of the part is covered from this. Fine. Okay then. Thank you. I hope you have enjoyed the session. So we'll meet in the next class. Okay.